Bom dia a todos. Agora nós teremos o simpósio satélite da GSK. Quem estará fazendo a apresentação será a Luciana Tarbes. Ela é farmacêutica, mestre e doutora na área de saúde pública e ela é gerente de Value Evidence da GSK Brasil, né? GSK Brasil, e ela então fará a apresentação do tema... o tema que está escrito. Desculpem. Então, vou passar a palavra à Luciana. Obrigado pela presença, Luciana.
Obrigada, André. Tá funcionando. Acho que eu vou ficar aqui, talvez, porque isso não vai favorecer meu salto. Mas, o tema, na verdade, é caminhos e formas de fazer parceria entre a indústria farmacêutica e o setor público. E aí a gente vai trazer um pouco a experiência da GSK Brasil, como que a gente tem contribuído com isso dentro do país. Tá? Esse é meu conflito de interesse, eu sou funcionária da GSK. Egressa da Fiocruz, Minas. Meu mestrado foi lá, então é um prazer estar de volta na Fiocruz.
Great to be back at Fiocruz to speak about science.
Bom,
well,
eu fiz aqui uma analogia.
I've made an analogy here of a plantation,
pouco do caminho
which is
da
a bit
clínica e
of the
desenvolvimento.
path
Se a gente
of
pensa do desenvolvimento
clinical
até
development
ao
and
seu
research,
lançamento no
the
mercado,
development
por onde ela
of the
passa,
drug until it is
porque
released
quais
at the
são
market,
todos os estudos e
what
todas
are
as
the
possibilidades que a gente
trials
tem de
conducted,
parceria.
the possibilities for partnerships.
Então a gente fala, deixa eu ver se o pointer vai funcionar. Não funciona. A gente vai falar desde ciência básica.
We're going to
então,
speak
como que a
about the
GSK
basic
investe,
science, how
traz,
GSK
funciona a
invests
ciência nessa área.
and
Fase 1,
incentivates
2,
science
3
in
e
this
4
area,
são
phase 1,
a
2, 3,
fase
and
clínica
4,
do desenvolvimento
which
do
are the
medicamento.
clinical phases of the
Também
development
não
of the
podemos esquecer
medication.
dos estudos
And we also
observacionais,
can't
que na
forget about the
realidade
observational
onde vai,
studies,
fertilizar, onde vai
which is
nascer toda essa
where
parte de ensaio
this
clínico. A população
clinical
vai ser
trial
usada,
part will be
qual
born,
incidência
what
que existe
population
dentro da
will
população,
be
qual
used, what is the
prevalência que
incidence
existe
in the
dentro da
population,
população,
how prevalent is this,
o burden que essa
the
doença
burden
traz ou não.
that this disease brings
E
or not.
como fiz aqui uma analogia de
And I've made an
que é uma
analogy
plataforma
here,
independente que a GSK tem.
the plants are being watered,
que
it would
eu
be
acho
the
que a gente pode chamar
independent
de
platform
fertilizante disso
that
tudo,
GSK
tá? Que a gente
has,
pode
which is the
financiar,
fertilizer of all of this.
ciência ou estudos,
So how we can fertilize the science and the studies
dessa produção do
and any of the phases of the production of this medication.
Começando aqui pela ciência básica
So
e
starting
fase
with the
pré-clínica,
basic
o que a gente
science
tem
and
efetivamente
preclinical
aqui no
phase,
Brasil?
what
Então,
do
a
we
gente tem
have here in Brazil?
programa institucional
We
da
have an
GSK, que fica
institutional
dentro
program
do
by
guarda-chuva de-
GSK,
do
which is
guarda-chuva de R&D, de Global Health.
inside our
Chama
global
Trust in
health
Science,
umbrella,
tá?
which is
Ele
called
é uma
Trust
parceria
in
que a GSK
Science. It's a
faz
partnership
de
that
forma
GSK
institucional,
has.
então a gente
It's
procura uma
institutional,
FAP
so
ou CNPq
we look
para
for
trazer
a
investimento. Então, a gente
CNPq or
investe
another
volume de
entity
dinheiro
to bring
e a FAP
investment. We
investe
invest an
A
amount
gente
of
abre os
money and our
temas de
partner
interesse da
invests
GSK,
another.
os pesquisadores
So the
submetem
researchers
no
submit
edital de pesquisa
this
seus
in a
respectivos
research
projetos.
open call. They
E
submit
aí a
their
gente começa
projects,
depois
and
de
that's when
aprovado
we
a
start,
parceria.
after they are approved,
Não
we
é uma
start the
parceria que a
partnership.
GSK
It's not a
simplesmente
partnership that
dá o
GSK
dinheiro. É uma
simply
parceria que a
gives the
GSK
money.
Vem, faz avaliação,
We
vê o projeto, trabalha
evaluate
muito
the
de perto.
project,
Então,
look at
a
it,
Fiocruz
look
já
at
teve
it
alguns
closely.
projetos
So Fiocruz
contemplados
has
no
had
passado.
some projects considered
Então,
in
hoje
the
a gente tem
past.
em
And
andamento
today,
com a
we
FAPESP,
currently have some
que a gente
projects with
investiu em
FAPESP.
10 anos, 10 milhões, e a
We invested 10 million in 10 years, and FAPESP another 10
O
million.
interesse normalmente,
The
tá? Esse é
interest
um
is
projeto
normally, this is
de
a
América
project
Latina, mas a gente,
for Latin
no
America.
Brasil é o maior investimento. E aí a
We
gente
have our
tem
greatest
como
investment here in
tema de
Brazil,
interesse,
and
tá,
our
normalmente
subject
dentro
of
dos editais,
interest
doenças
in the open calls are
negligenciadas,
usually
imunoinflamação,
neglected
oncologia
diseases,
e HIV. No Brasil, a
immunoinflammations,
gente
and HIV.
contemplou mais,
In Brazil,
isso
we
foi a demanda, não foi a
considered
gente,
these
porque o
more
edital
because
era
of the
aberto.
demand. It was
Doenças
an open call.
negligenciadas, muita coisa na
We
na
looked
área
at
de
neglected
leishmaniose,
diseases,
vacinas.
leishmaniosis,
E,
vaccinations,
geralmente, a América Latina tende
and
mais a
usually in Latin America,
Esse projeto
immunoinflammation.
começou no Brasil em
This
2012
project started in
e já
Brazil in
foram,
2012,
cerca de 24
and we've
milhões
had
investidos.
about 24
pelo
million invested by GSK.
Quando a gente passa
When we
de
go
novo,
on
desculpa, dentro do guarda-chuva do Trust in
within
Science e
this
Global Health,
umbrella of the Trust in Science and Global Health, we are expanding our partnerships. So beyond the financial partnership and evaluating projects by GSK Global, this is our global investigator that helps us with the Trust in Science program. This is an article that was out in Nature. And we also signed a partnership between FAPESP and Bio-Manguinhos to support projects that are startups, because we came to the conclusion that us researchers know very well how to do science, but we don't know how to sell our projects to the industry. So how can we make this partnership happen? GSK entered in the technical area. How can we look at the investigators in the basic area, help them technically, if this project is interrelational, if it's following the technical part correctly? And Bio-Manguinhos supports the business area, how to sell this project, how to show it to the market and other pharmaceuticals. And finally, at the end of last year, we signed a partnership between FAPESP and the British Embassy to bring 2 Nobel Prize winners. We don't have the date defined yet because they have a very busy schedule, but there will be a great event to incentivize, to understand what their path was to win the Nobel Prize in Science, what was the path they made in this research. Now, going back to our plantation, when we get into the clinical phase, we're financing a lot of basic and preclinical elements, discovery of alpha therapeutical molecules. Then we go on to the clinical part. And we can see in the trials in Brazil, we conducted research. The normal standard in the Brazilian environment is phase 1, phase 2, and phase 3. These are normal in the area of oncology and HIV. Phase 2, phase 1 less normal. A lot of investment in phase 3. And phase 4, you can see a progressive increase, progressive observational increase here. What we observe in GSK is that this model repeats itself. We have high investment here in Brazil in phase 3, which is very good because we already offer this access to medication to the population. We test the medication on the Brazilian population. We engage the researchers in global projects, and usually these projects are multicentric. I think most of you are familiar with this. They are multicentric, so there is a global protocol with, you know, what the kinds of patients are, the characteristics that will be included, and Brazil is included as a center in which this protocol, we translate the protocol and enter this into this study or trial. For these kinds of trials in 2017, we had more than 150 research centers and units involved in our trials, more than 150 Brazilian researchers involved in our investigations, and more than 2,100 subjects or patients recruited in these trials. The investment in Brazil in 2017 was around 30 million reais, and globally in the year of 2017, 18.5 billion reals. When we go on to the observational studies, and this is very particular here in this country because we're speaking of how the system works, how the population is distributed, how this disease or illness happens in your country, we have more autonomy relating to the protocol because it's what we need, what the ministry needs. So what are our demands? So we can proactively submit protocols to the global team. So we compete with the whole world in order to get our financing. It's like a foundation that supports our research, and through this we can design the protocol, make suggestions, say why we need this, and start the trials. So most of the time these are the models. They are transversal. We do a lot of transversal work, a lot of descriptive ones, trials for the use of medication, and also studies relating to the costs in health, because this is all very specific to our country. The areas in which we are most interested in are the respiratory area, lupus, vaccinations, and oncology is green here because GSK, RNG of GSK, the RNG of GSK is changing its route a bit. So we sold the part of medications that we had already launched in 2014. For oncology, we continued studying this oncology pipeline, and now is the moment to relaunch molecules. We are investing greatly in this oncology area, so most likely we will anticipate and start observational studies in the oncology area. These numbers are for 2015, 2017. We had more than 40 publications. So this is a bit of a smaller structure in Brazil, more than 40 international and national publication with Brazilian data, and about 30 million reals invested in this kind of study. And then we have this part of watering the plants. It's a platform that GSK has launched. It's a new platform format. As I said previously, it can be in any of these phases of the clinical development. Today, previously, well, this platform always existed, but I will speak more of this later. We do more of a certain kind of proposal in these areas. We speak about asthma, lupus, HIV, oncology. What interests us in these areas? So the researcher can submit to this platform. Once they submit, this goes through a national evaluation board. Once it passes this board, it goes to the global board. The global board evaluates the study. If it is approved, the person receives the financing, or it might not be money necessarily. It could be a medication. It could be a support for this publication. Doesn't have to be the whole project, so this platform is a lot more flexible. We have 9 approved studies last year and this year. We have them in HIV, asthma, and lupus. Lupus in the immunology area. This was our pilot area at GSK, and that is why we have more approved studies in this area. However, currently we have launched this platform that is open. So it's the supported studies program by GSK, and you can find here, these are the areas. You can click on each area and you will have the related subjects there. So we are really connected to the global team, so the material has to be in English. It has to have the quality, a global quality of science, because we are competing with the whole world. But fortunately, we have had great results. From 3 that were submitted for lupus, 2 were approved. And in the last open call of 7, 4 were approved. So this is good recognition. And this is a way of obtaining the financing. The advantage of this platform is that GSK cannot interfere with the study. We are not the authors of this study. We cannot interfere with the method. We can make questions. So as the financer, just as FAP, you can do this. You can say, I don't understand what's going on here, but we don't interfere in the objective of the study. And it is not necessarily connected to our products today. This is a platform that will have a partnership with our pipeline that I will show. So it's a way for Brazil to get ahead in the pipelines and to build this future with GSK. So inside our pipeline, here we have great investment in oncology, HIV, immunoinflammation, respiratory, other pharmaceuticals, which is a minor part, and vaccines. Here I brought just a brief summary of what we have in terms of volume of pipeline and assets. So in oncology we have 16, being that most are in phase 2 in oncology. We have a number of molecules that are approved in phase 2. So this is phase 2, HIV, 11 assets, 3 that are being launched, 4 in phase 2. In immunoinflammation, it's more distributed, so we have lots of things in phase 1 that will still grow, and 4 in phase 2. And in respiratory,
we have 10 assets, 10 molecules being studied, and here are the 4. And in vaccines, we have 17 molecules under study, but the majority is in phase 2. And what I brought here, that probably is not a very well slide for you to see, but this content is easily found in Google. That within oncology, our priority in short term, let's say, is multiple myeloma and head and neck cancer. That's what I would like to call your attention. And in terms of vaccines, we have approval in adults. And now we are having studies in children for Becero, and the essential virus, that is a platform that is of great interest, and we include children and adult populations and also mothers. And that's it. Well, our mission is to invest in science, and we've been doing that for 110 years and building science in Brazil. Here I focused in R&D, so we have a little bit of strategies, which is not the focus here, but we have an innovation program launched in SJK, in which we created this program called Together, and we opened partnership not necessarily in R&D, but with the 10 challenges for startups. And yesterday we launched another program to generate data in this innovation umbrella of GSK, and we called Data Tool. This is a hackathon of data tools in terms of epidemiologies, and I think it also links with the Innovation Hub, and we are learning, and our IT director is here. She is responsible for the innovation program. So that's it. Our president is here, and I hope we have 110 more years contributing and investing in science in Brazil. Thank you.
Alguém tem alguma questão?
Do you have any questions, any comments to say to her?
Comentário?
Anything to say?
Bom dia. Eu só queria saber pouco
I
mais
would
dos
just like to know a little bit more, because you talked about vaccines and products and some diseases that may be neglected, but are they all focused on biologicals, or you also have synthetic molecules?
Temos dois. Provavelmente distribuído de forma
Well,
diferente
we
entre
have
as áreas.
both, probably distributed differently. In oncology, we'll have a lot of immune and biologicals, but in respiratory, we'll have synthetics.
Está em transmissão.
Bom dia. Geralmente o projeto para
Good morning. Generally, the project to be approved and evaluated, how long does it take for you to evaluate the project? The project that the person submit.
O André resolveu. 98.
Well,
bom, vamos lá.
Let's see.
Transparência.
Transparency for pharma, because GSK is quite huge, so we have different structures. For pharma, as we call, that is the part of oncology, except vaccines and HIV. So pharma, 98 days to have the result. For vaccines, you follow the flow, so there is no deadline established. And HIV is much faster. Generally, it's faster than 98 days. But if it is in the case of basic research, the person will submit the project, and we don't know if the project will launch or not. So how do you evaluate that? You send to a person to understand the viability of the project, or if there is a risk? Well, we evaluate it before. As it is basic, we don't have technical knowledge in Brazil, so it will be evaluated globally, the viability and interest. If it is via the Trust in Size project, it will be between FAPESP, for example, and GSK altogether. So generally, they invite ad hoc researchers to be part of the evaluating team. Any other questions, comments? No. So, Luciana, thank you for your presentation, and thank you. And right afterwards, we'll have another satellite symposium.
Isso, Você quer que eu apresente?
Você quer adiantar?
Mas depois, na hora do... Só para convidar os próximos.
Ah, sim. Deixa eu ver, o próximo é as 10, né?
É Patrick Gertin, da GE, falando sobre inovações em vacinas e produção de viral de vetores.
Acho que a gente pode dar esse intervalinho.
Isso.
Teremos pessoal, bom dia, teremos breve intervalo.
Well, we'll have a small break, so we come back at 10 with a GE session. With Patrick Gertin, who will make his presentation on new technologies for vaccines. Thank you for being here.
Depois esse negócio poli cai.
Obrigado, Luciana. Muito bom.
Obrigada, André.
Desculpa qualquer coisa.
Não, mas...
É que tá um pouco corrido aqui, que a gente tem também limite de
But I think that's why we need to continue to move forward on different therapies, gene therapy, cell therapy, viral vectors, you know, in anticipation of what could come next. Because just like, you know, any form of nature, right, the evolution and adaptability, you know, we're evolving all the time, and the diseases that are challenging us are evolving over time. So I think that looking at these new mechanisms, you know, and how we use them, you know, how do we use a specific delivery system? I mean, if you look at some of these features on the right, components on the right, you know, they're really elegant mechanisms on how they can act with your body's cells. And so looking, you know, just on the viral vector specifically, looking at from a 2016 study, looking at some of the numbers in the clinical phases, you know, we look at a total of, you know, 726 at that time, and proportionately how they're represented, the largest percentage of those being the adenovirus, you know, followed by the retroviruses, so on and so forth. But you can see the relative breakdown. And so, for example, if you look at all the products, both therapeutic and viral, in the clinical phases, you know, whether it's, you know, phase 1, phase 2, phase 3, what have you, you can see over time, since this period and even more recent studies, that percentage is increasing. So the total number tends to increase overall in terms of a trend, but the proportion of these respective components also likewise increases. So, you know, if we look at vaccine production today or in the recent past, we look at, you know, processes that were really developed decades ago with older cell substrates or eggs, limited purification. You know, they were processes difficult to scale up with certain laborious steps involved in the processing. You know, unfavorable process economics, some of the increased regulatory requirements. And so, again, if we look at each of the components within these categories, we see that, you know, the egg-based production or other cell types are being replaced by plasma cell or more regulatorily accepted. So when you go for your filing, you know, you have an easier path to it because you're using a well-characterized cell line that has a very rich history. So again, this is a critical component. You know, where before it was very expert driven, you know, now there's efficient purifications based on chromatography. You know, again, science and art kind of coming together, so to speak. So the scalable technologies and the single-use technologies are certainly playing a significant role. And, you know, I can tell you, and not to digress from the point at hand, but with respect to some of the single-use technologies, I've used those for microbial fermentation, mammalian production, you know, subunit vaccines, virus-like particles, traditional viruses, vaccines, et cetera. So again, the point is, you know, one should try to use technology that's adaptable. Again, as I said, for what we have for today, but what we anticipate for tomorrow. And then with respect to the unfavorable process economics, you know, efficient and rational process design, we'll talk about it more in a moment. You know, the QBD, or quality by design, continues to be quite essential for the rapid approval and forward movement of any process. And really that starts from the beginning, right? You know, so before you embark on a particular project, you know, use the tools that are available, use the literature that's available, you know, talk with people, team up with other collaborators who have knowledge in the space, you know, and really take value, you know, contribute to them and accept help from them. So, you know, closed handling, closed processing, as you know, always the environmental considerations are always a challenge. Closed processing, especially in the recombinant protein space, has really moved forward. Again, that's what allows a lot of ballroom settings rather than traditional room-by-room segregation. So things like connectivity, closed processing, significant filtration applications, these are things we should think about. And then, you know, back to the quality by design. I mean, at the end of the day, you know, all these things, you know, it's tremendous knowledge, but really we know what target titer, what volume, what product per time we need to produce. We also know what are the critical process parameters by which we have to operate our machinery, and ultimately what is the critical quality attribute, CQA, of the molecule and some of the contaminants, you know, things like clearance of host cell protein, host cell DNA, these types of things. And, you know, where there are still many processes with serum, I think they're becoming less and less over time. We see the chemically defined media. So, you know, again, this is a reiteration of the earlier slide. I won't belabor the point, but one of the a single-use platform utilization, this happens to be our virus production, you know, moving from left to right. If you look at the molecule design, the cell design, selection, cell culture, you can see that, you know, it really starts with the cells and the constructs and the bioreactors, the clarification, the downstream considerations, that space, and, you know, how efficient that process for your molecule. And we always like to think of our molecules in particular, you know, from a very important perspective, right? That's our company's business, that's our company's, our future. But, you know, again, talking about the earlier point about flexibility, you know, there may be a point when you want to use certain machinery to produce analytical components for the testing of your molecule. So again, if you wanted to produce a series of antibodies for assays and do that in-house, you know, if you had the equipment and the right know-how, you know, that can certainly benefit you. And so, you know, taking a look at some of the single use in the vaccine production, you know, oftentimes manufactured in small batches. So, you know, in that way, single use is appropriate. You know, the campaign manufacturing is important. And again, by no means am I anti-stainless steel. You know, again, I have 30 years in the industry, 16 of which was in stainless steel and glass, 14 of which is in single-use technologies. And I can tell you that each one of them has their place in their application. You know, it's really how do you choose to use it and what's right for your process. You know, and I've always said as well, you know, if you've already invested in a lot of technology, you know, don't necessarily abandon it, but learn how you can hybridize or modify and make more flexible the different unit operations. You know, the third bullet point is a really important one because of the pandemic preparedness. We did a contract a number of years ago. I can't talk too much about the details, but again, it was in pandemic response, and it was about generating, you know, 30,000 doses in less than a 6-month period, starting with nucleic acids. So again, it's really important to address any of the market needs, whether it's pandemic response, whether it's endemic or what have you. You know, some of the high cost constraints, you know, with vaccine, you know, need to have process improvements. And if it's in a single unit operation, that's still going to bring you a lot of value. I think a lot of people philosophically think of things, and listen, you know, we're only human, right? So we think of things in an all or nothing fashion, but it doesn't have to be an all or nothing fashion. Okay, so, you know, as I've always said, you know, just to keep the mind open and see, you know, what are other people thinking the industry doing, and what are my needs? And safety in the cross-contamination is an obvious one. So the single-use processing, the closed systems, you have a single-use bioreactor on the left, single-use connectors, and the ready-to-process column. And again, one of these considerations, you know, really, again, is the, you know, negation of certain infrastructure, clean steam generator, WFI still, you know, these types of things take tremendous energy and money to maintain. So again, taking a look at some of the compiled data from a 5-year period previously, substantial savings with single-use technology. We see the first column. I won't belabor this point, but really, you look at each of the respective categories, and with the single-use, consumables is always going to be your largest cost. The capital facility cost is going to be lower. The labor is generally going to be lower because, again, with some of the closed processing or single-use ops, operators can do dual roles, don't have to be necessarily segregated to a single role. Is my sound okay? Am I okay? Yeah, okay. Sorry, I can't tell really from my perspective. But, one of the most significant ones, and this is more of an environmental consideration, but I think we do have to be environmentally conscious while we're doing this manufacturing, is, water utilization. The water and energy utilization in our analysis over time has really relates to about 75 to 85% less water use. And a lot of that comes from the absence of CIP or SIP for respective unit operations. But the main point is, as water becomes more and more precious, I think that if we can achieve our goals for manufacturing in an environmentally conscious way, not only is it a cost savings, but I think it's a philosophical advancement. So but the bottom really just shows some of the unit ops in terms of mixers, skids, reactors, and purification skids as well. So for the adenovirus vaccine production, just a schematic showing the scale up from left to right, what's entailed in the downstream. And on the right, really is an important component of it is really the analysis, okay, because we have the steps for the unit operations, but, a key variable is the quality metrics and what testing's going on throughout. And again, what's the timing of that testing? Can you do real-time analysis, or are you going to be delayed? So, the cell substrates for vaccines, if we look at the production system on the left, really showing more of a traditional cell platform, on the right, more bacterial, showing the vaccine types, whether it's attenuated, inactivated, SUV, VLP, so on and so forth. On the right for the bacteria, the conjugated polysaccharides and things that, again, can oftentimes have challenges from a downstream perspective. But the main point is that it's really a broad spectrum that we have to choose from. And as you're looking at the cells, things like, again, what's the history of the cells? What are the characteristics of the cells? Do you get a higher titer from a particular cell strain that happens to be attached? So you have a little extra challenge for the attachment, but you gain the benefit from the overall titer. So, the cell substrate, a lot of the ones nowadays that are really preferred are, the Viros, the MDCK, the duck cells, so on and so forth, good safety track record, animal origin free, good virus propagation. The media available, again, I think it's important to keep it animal component free wherever possible. And looking at the adherent characteristic, as I mentioned earlier, versus suspension cells, cells like, Viro, for example, are attachment dependent. For example, HEKs can be attachment or suspended or suspension. So I think it's important to kind of balance, what works best for you and really what cell line results in the best quality attributes for your virus or vaccine. The feature about the attached cell lines as you embark on those is, it is a more complex scale up. But again, if you're getting, 10x tighter, then that's value. That's worthwhile. So looking at some of the downstream components, in full disclosure, most of my career I've been upstream, bioreactors, et cetera. I have done some downstream. I don't tell my downstream colleagues that I've done that because they go, really? But I have done enough to know a little bit, so sharing some of this. But again, with the clarification, first step, filtration, normal flow filtration, TFF, centrifugation. If you look at centrifugation, I mean, there are some single-use centrifuges nowadays, but most people, for the economics and the relative centrifugal force that you need, will go with a traditional, but again, that encompasses cleaning. Okay, so you have the capital cost of the unit, and you also have the CIP. Looking at some primary capture, focusing really on the fourth point down, the chromatography, IEX, mixed modal, AC, affinity chromatography, hydrophobic interaction, so on and so forth. There's also the last bullet, the membrane platform, which can also be very effective. So when you think of these things, you need a particular step. Again, whichever step, whether it's starting out with an affinity step, followed by an anion exchange, followed by cation exchange, etc. But it really comes down to the other decision. Again, how are you utilizing that resin? What's the resin lifetime? Are you using a traditional glass and stainless steel column? Bit about that. Excuse me. We'll talk a little bit about that in a moment. About some of the labor costs associated with packing a column. And also some of the qualifications of those columns where, in certain instances, you can put that burden on the vendor or manufacturer of that unit operation. So, with some of the core bead technology available now, again, essentially, this is a modern alternative to SEC. And really what it is with the technology, it's an agarose bead, but rather than just having certain divots or compressions in the resin or surface ligands. It's really channels through the beads with the ligands attached on the interior channels of the beads. And what this actually does is this increases the speed and efficiency and increases your total surface area that you're able to use. As you can see schematically, we have the virus molecule, the largest component on here, the fragmented DNA, the host cell proteins. So again, this would capture the contaminants, allow the and so with the core bead application, there's also, especially for some of the viral work currently, Capto-Devirus, which is actually another agarose core type of bead, but with the sulfate-like ligand on the interior channels. So the point is that this change to some of the resin design has actually enabled the increase in efficiency and speed. So, again, is it right for your molecule? I think you really have to test it and see. So, taking a look at the applications for some of the core beads, you can see a pretty broad spectrum of molecules, virus, et cetera, that can be applied here, even VLPs. I think there are some commonalities among all of these components, but again, there's also unique differentiators and also challenges to reduce your contaminants, whether they're product-related or non-product related. So, a cost breakdown, some of the steps in the simulation alternatives, looking at the literature search. Again, as I said earlier, check out the literature, see what's being done out there currently. Talk to your colleagues within your team what they're doing. There's also, with the process modeling from a very powerful tool. Set up different process alternatives, and then evaluate results and identify the economically feasible unit ops. And as I said earlier, not taking an all or none philosophy, where does a particular, you might get from the replacement of one single unit operation, you might get a huge benefit, okay? It might save a day in processing, and if you get X number of batches a year and you save a day in processing, you might get an extra 1, 2, 3 batches per year, and again, that's really significant. And then, again, process development, it has to be done. You can do all of the other things in terms of modeling and literature searches and communication, but it really comes down to the process development, to really evaluate these components. So if looking at some of the process alternatives, we look at 8 different scenarios here, starting on the left. upstream process, detergent treatment, upper portion of the bracket, nuclease treatment, or lower the clarification with NFF, TFF. And as you can see with all of them, I won't bother reading. I won't bother reading each of the respective. Sorry about that. That's actually my alarm. It's not my phone. No, I wouldn't allow the phone. The alarm, yes, no phone. But what we see here really is a comparison of 8 different process flows for the same molecule, for example. And really, again, I'm going to show momentarily some of the cost per gram to produce and apply this this model or this evaluation. But again, it comes back to the primary component that I described earlier. How is that for your molecule? How is that for your process? What are your acceptability specs? The quantity of DNA or quantity of host cell protein per dosage or per mg. Okay, so you might get a process that is apparently economic according to a modeling exercise or according to one molecule's evaluation. But again, if that's for molecule A, if you apply this to molecule B, it might be a little different, right? So again, everything, some things that might cost less comparatively, you might not still be the top performer. So again, you may have more challenges at lower cost. Okay, so again, it's always important to look at this. And if we look at the cost per dose of the different bioreactor scales, this is a schematic representation of the cost per dose on that last exercise that I just showed. And as we can see that, comparing one to the other, and one of the main features of this slide, okay, there are subtle differences between each one, but I think from back to front, looking at 50 liter to 200 liter to 500 liter, I think what you realize, and a lot of people know this quantitatively, and a lot of people know this intuitively, you get a economy of scale, okay? Because of some of the materials and some of the operational logistics. Again, you really are gaining financially from the scale up. And then, looking at some of the contributing cost factors for each of those respective models. If we look at it, for the royal blue color, the capital, a lot of times, you'll get a higher cost if you have something like, the certain stainless steel resins or housings, depending on the cost of those housings. You have components like materials, obviously represented by the dark gray, and those tend to be largely equivalent. The consumables cost, again, where you'll get that's the light blue color in the center, and really where you'll see that for the consumables cost is if you have things like membranes, for example, again, because that's representing, something that's being utilized, as a consumable. The labor, if you have a traditional column, you're going to have to pack that. You're going to have to qualify that. So if you look at the darker portion, the dark navy blue color, again, the labor for packing, qualification, that's just sort of a very simple example. And then there are other extraneous costs. But if you look at the total, sort of varying, US dollars per batch, varying, somewhere between about $55 and about $85. So that's about a 35 per batch difference. But, again, if you take any one of these steps, if we look back at, here, if you were able to replace one component in that flow chart, again, it could result in significant benefit. And then just the evaluation of productivity. Really, the core technology, this is just an up-close look at the core technology replacing an SEC step. And again, not necessarily replacing every single step, but just for the SEC step, and showing that with the respective matrix and the channeling and the ligand attachment to the interior channels of the beads, you can certainly gain benefit. And, just showing, and this is intuitive to most here, the principles of SEC with the higher molecular weight components being excluded from the pores, coming through first, and then proportionately according to size and hold-up time through the system. And the productivity, if we look at the, again, economy of scale or product per time, we see that the core bead really, especially at the 2,000 liter scale compared to the 200 liter scale, certainly outperforms. And then if you see on the left, the core bead technology in a stainless steel column, again, certainly increases above the traditional SEC and stainless steel column, but still slightly behind the single use. And then, in conclusion, the paradigm shift for vaccine production from lab bench process to rational design, incorporating process economics, combination of single use and traditional. I think those are really good things. It goes back to our original discussion, right, about staying open-minded and potential hybridization and what works for a particular unit operation. And then, again, looking at, core bead technology, it may or may not be right for your process. We've seen it in most cases that it is absolutely right for a lot of processes. But just in the case of, for SEC, how does that apply itself? And then I would like to thank the team shown here, especially Mats Lundgren, who was significant in putting this presentation together, the Uppsala R&D team, who are terrific. Again, the Marlborough, Massachusetts, just outside of Boston team is very good at the vaccines as well, but for purposes of a lot of this study information today, the Uppsala team. And again, that goes back to one of the things I said earlier about the different teams. No matter where you're at geographically worldwide, bond with your team members, try to find out what they're doing, try to support them, try to get support from them, and just really, embrace the collaboration. So thank you very much for your time.
Vocês têm alguma pergunta? Do you have any questions for Mr. Dr. Patrick? Any questions? Alguma pergunta? Algum comentário? Não? Nenhuma pergunta, nenhum comentário? Certeza? Então, mais uma vez agradecendo a palestra do Dr. Patrick. Obrigado. Thank you very much for your presentation. E nós teremos em seguida outra sessão, então faremos um breve intervalo e em seguida teremos outra sessão. Thank you very much. Obrigado. Ok, thank you.
Para discutir inovação no âmbito de Bio-Manguinhos, e teremos hoje as apresentações dedicadas já à apresentação dos ativos de Bio-Manguinhos especialmente, né? Ontem a gente teve a abertura com a apresentação da cadeia de inovação da Fiocruz e de Bio, e hoje a gente vai começar a falar especificamente de Bio-Manguinhos. Teremos então a abertura com a apresentação da vice-diretoria de Desenvolvimento Tecnológico de Bio-Manguinhos, que vai ser feita pelo Felipe Luca. E, em seguida, temos as apresentações do gerente do Laboratório de Experimentação Animal de Bio-Manguinhos, Rodrigo Miller, e depois, em seguida, a apresentação do gerente do Laboratório de Neurovirulência do Dr. Renato Marchevski, de Bio-Manguinhos também. Agradeço e passo a palavra Felipe, por favor.
Bom dia, bom dia. Vocês estão me vendo direitinho?
I am Felipe De Luca. I work with Sachiris and all of the rest of the team of the Vice Directory of Technological Development. You're not hearing me? Okay, I think it's working now. So because Sotiris could not be here and Marcia couldn't be here as well, so they asked me to make this presentation. It's quite short. We're going to speak about drug discovery platforms, but actually we're going to take a step back and say why we're thinking about platforms and services. That's the idea of the presentation. Let me try this out. Okay, so the first thing that we should consider in this context is the regulational conditions that we used to have relating to innovations in Fiocruz and Bio-Manguinhos. This changed recently because of the technology and science milestone, and then in 2018, based on this milestone, Fiocruz launched the innovation policy, and after this, the Fiocruz units, including Bio-Manguinhos, started thinking about what business models they could use, which would be possible considering the internal structure and the know-how that we have. So that's basically it. This was the alteration in the regulational environment that happened and motivated this presentation. So the first idea, considering this milestone in the regulation and this change in the environment, was to think about what our capacities would be, what are our positive characteristics in Bio-Manguinhos, and what we could use. So VDTEC, which is the Vice Directory of Technological Development, other tech labs, micromolecules, bacterial, biosafety. So there is a group of labs that are a part of this Vice Directory. And along these years, they had the opportunity to work with or make partnerships with great pharmaceutical companies like GSK. And this experience is very positive. It's an advantage on our end. We can use this, not just the knowledge that is shared during these collaborations, but also the mindset of these companies was something important. Something that made these partnerships important. And this is something we considered and we are currently considering. Let me just go back here. Okay. So that's it. We have our internal structure with great experience, great thinkers and researchers here, highly qualified people in here with a lot of experience in partnerships with big companies for producing and developing new pharmaceutical products. And so there's a piece missing here. What does the market need? That's basically the project, this initiative of platforms and services that the Vice Directory wants to get to and wants to offer at some point. We will count on these steps. First, we need to conduct an analysis on the market. So the idea is to work from business to business. And so to collaborate with other companies and convince other businesses to pass their innovation cycle through the Bio-Manguinhos structure. And so we need to know what they need. The first idea is to analyze the market, assess the market. And then, considering the experience and the know-how that we have, look at this and compare what the market needs with what we have. So where we are and where we want to get to. That's the idea, and along with this we will identify gaps. The idea is to conduct an internal reorganization to eliminate these gaps or at least reduce them. And with this, we can try to find business models or services or platforms that can attend to this initial demand by the market. It's pretty simple. Basically, this is something we started working, and it's what we're doing. If you take a look here, it's all very recent. This happened in 2016. After that, Fiocruz started thinking about how they could work with this regulatory milestone. Then October 2018. So this is still in a preliminary phase. We have a structure of what we would like to do and an idea of how we want to do this, and we're walking in this direction. So looking at the vision that we have today, which is still very preliminary and superficial, we can already see some values or advantages, that competitive advantages that could work in the future. The production of eukaryotic cells, viral production, the development and production of monoclonal antibodies, the recombinant proteins in E. coli, the sequencing of cultures, the increase of scale in bacterial culture, and even consulting for biosafety. So this is what we have today, but it's still very preliminary analysis. This is the future. So from this market analysis, we need to look at everything that can transform into a competitive advantage, and we need to make an effort to diminish these gaps and then launch these platforms, one or more service platforms that can serve this demand from the market, and be able to create partnerships from this and help other companies to deliver products that are innovative and can contribute to our objectives, Fiocruz's objectives, public health objectives in Brazil. So that's it. Thanks. I told you it was going to be short. Thank you. Thank you, Felipe. So let's leave all of the questions for the end, okay? So Rodrigo Miller. So I'm just going to get started. Good morning, everyone. Can you see me? Okay, great. So we want to present the technological services that we can deliver today. What are our portfolios, services that we offer? Today we have internal clients, especially EVDTech. We have experimentation that already offers internal services for all of EVDTech, LATEV, and others who are also here. So we have cutting-edge technology, and we have our technological partners that we have used for preclinical trials for products, and in these partners are GSK. We have a huge infrastructure. I'm not going to get into all of the details, but the operational area is almost 1,000 meters. We have a special air conditioning system for the animals. We know that trials with animals today are very important in their specificity, and the data for reproducibility, it should be considered this so that the study can go on. So we have a team that is highly trained. They are certified. Internal certification has been created so that we know everyone is trained and specialized in what they do. There are not people who go into this. We have the internal control. We have 11 technicians, administrators, and all of these people in the lab. We work with rats, rabbits, and guinea pigs. We have activities for quality control. and things that our control department utilizes. We have technological development, as Felipe mentioned, some areas that are providing external support, and technological upgrading and transference. For the unified health system, we work with good laboratory practices. This is a frequent qualification for our professionals, training in biosafety, consulting in infrastructure. For other locations, also inside Fiocruz, we have this consulting for structures and the experimental refinement for our clients. If somebody seeks us out, we conduct this consulting to improve the studies, the trials with animals, so that we can obtain a good result. And this is something that is very relevant today. The quality control, we have non-specific toxicity tests, pyrogen tests, stability tests, potency tests, virulence tests, and reactivity tests, skin reactivity tests. These are all tests we conduct for quality control. Also, the production of anti-ovalbumin in rabbits. We have technological development, lots of activities here relating to recombinant protein, quantification, bacterial quantification in animals, passive immunization, evaluation of vaccines. Today we know there are many difficulties to find the best vaccines and get the best responses. The antibodies, murine antibodies, production of monoclonal antibodies as well. And the methodologies that we use, there are a variety of experimental techniques. We do a lot of things and are constantly improving our techniques, and we are always in contact with our researchers in order to improve these experimental results and make them efficient. So with the mice, there are different pathways. We have cardiac puncture, saliva, etc. In the guinea pigs, we also have blood collection by cardiac puncture, and the rabbits as well. And we also work with chicks for the production for the blood collection. So there are a variety of items. I can't speak of everything right now.
We also have subcutaneous inoculation, endemic, intranasal, intrabrain, in neonatal individuals for collection as well, in guinea pigs as well, and also in rabbits. So from 2014 to 18, we had almost 300,000 experimental tests. So we have repeatability and every employee coming and being trained so that we can reduce the number of animals, because we can have a specific technician working for that, and we know each one of them and their individual performance and how they work in the animal tests, and they have to be the only ones in the room. We generally have only 2 people in the room due to odor and noise and so on, so that experimental test and the responsibility of people there are quite huge, so that the test is effective. We know that from preclinical to clinical trials, it's a difficult process, so in our lab we have to make it as perfect as possible so that we can evolve. Now we have 15 immunobiologicals under development in our lab. Some of them are really there. Every year we have about 700 trainings in the lab with our employees, and we have 12 there in the animal areas, and we have 700 trainings for them every year, and this never ends, it's cyclic.
E a gente tem programas de qualificação.
And we have qualification programs because it's not only people. We also have equipment, and we need to check the areas and the scales, because if we weight different grams, we have a different result. We need to be everything to be proportional. So we calibrate every equipment, and people, even the air conditioners, are checked so that the animal experiment may be effective. So that's what I wanted to tell you. Thank you.
Chamo então para sua apresentação, Dr.
Now I
Renato
invite Dr. Renato Machevski for his presentation. Well, good morning. I'm sorry some of you are standing. There is a seat only for a short time. You may sit here. Anyway, at first, in honor of two architects, Luiz Moraes Jr., who created Bio-Manguinhos within a unit that you know is important, but secular Fiocruz is quite important, and the architecture of Luiz Moraes, and here Santiago Calatrava, who built the building that he says everything is curved, and I was somehow afraid of entering here, and obviously, you know, it's a pleasure to be invited to be here in the Museum of Tomorrow. First, I would like to congratulate my colleague Rodrigo Miller, and you can see he is quite thin, and it shows the non-stop activities he does. Laneu, and I'm part of this group. It started in 1982, and I have only 58 protocols with non-human primates, so you see it's a huge difference in the amount of Rodrigo has shown, and you can see that working with 58 protocols, it's a lifelong experience. So I would say that ideally, no monkey or no non-human primate should be used in research, but we have no alternative. We are obliged to use them. But our only idea is to have scientific development with the use of non-human primates. So I won't say I refuse using them, but in Dr. Akira's dreams, within this time I would be in Manguinhos, non-human primate would be used anymore in the end of my career. Unfortunately, it's not a reality.
Isso é para relaxar.
This is just a quick time for you to relax, because I see something incredible. I don't see any glass of water for a hypertensive and diabetic person. I can see a bottle of water there, but I don't want to take your water. No, please, take your water. But you are responsible for the invitation.
Então vamos ver se eu aprendi. Não é que eu aprendi? Bom, vocês estão vendo.
Well, as you can see, we had the first problem here when our colleagues said that they were managers in the areas. I believe they should check that if Vivian and the reviewers would make this kind of verification, but 1 month later nobody corrected anything, and we are receiving our material in the last minute. So what do we talk about animals? That they shouldn't be used. You all understood the message. The second message is that without them, we wouldn't have all the developments we have. They are unavoidable. So when we say we use non-human primates, at least in Fiocruz by EOCD since 1909 with Carlos Chagas, we understand that we cannot do anything in the medical area without them. So the idea is that monkeys couldn't, in fact, not be used. And the proof of that is that every year, and I have data from 2018, 100,000 non-human primates are still used worldwide, showing that despite all the ethical part involved in it, we still have this kind of work. So we bring this idea that we have to use them unavoidably because all development depends on the use of non-human primates. Despite Rodrigo being the owner of a lab using rodents in an incredible number, and he didn't mention this number, but you would be shocked on that, we still have today only 0.5% of researches on animals with non-human primates. If you take a look at these numbers, you will at least believe it's just a few, but 100,000 in the world is still the guideline, and this progress cannot be prevented. Another very important part of our history is understanding something called divergence. When we use, for example, a chimpanzee or a bonobo, that is the good chimpanzee, let's say, it is the one that is not aggressive, we have a difference with the different ancestors that may be 8 million. There are some controversy in this data regarding, for example, the monkeys. We don't have the synalizers here, but the ones that we use a lot in the research lines, that is rhesus monkey, we have a distance from the common ancestors of 25 million years. It is translated in a noble part of history that is genetic homology. The closer to the ancestor, the more homology we have, more physiological characteristics, more characteristics making experiments closer to the ones carried out in humans.
Houve mais uma trava aqui. Ótimo. Agora foi. Dividimos os macacos em macacos.
We divided monkeys into monkeys from the old world, the Africans and Asian ones that are really the most substantial form used in experiments, and we have two representatives. We have the rhesus monkey and the cynomolgus, that is Macaca fascicularis, also known as the one who eats crabs. And those monkeys, in fact, from Asia and Africa, are the core of our scientific experiments. And we also have the monkeys from the New World that, for some interesting reasonability, they don't respond very well all the time because their phylogenetics is distant from us. We have about 35 million years from the common ancestor, so a chimpanzee 6 million years apart. If we think about a gorilla, 7, 8, 9 million years, but when we think about the rhesus, 25 million, and when we work with neotropical monkeys, 35 million years, or even before, in terms of comparing to the common ancestor. So it shows a genetic homology that is not the same. Although we have the genome that is similar, but we don't have 93% of homology. This is a noble sign for you to know which experiments may or not be similar to those carried out with the characteristics in human beings. This relation is changed today into a kind of a villain because thanks to the genome research, basically all animals from the new and the old world are now close to a definition, favoring scientists to find out which are the best biomodels to be used in the future. In our unit in Fiocruz, the ICTB unit, which is the Institute of Science and Technology and Biomodels, and they are responsible for the production and provision of biomodels. We have there, and I put here, if you work from there in ICTB, probably you will correct me because, again, the review was not good. We don't have Colitrichs jactus, but they have Sairime scienturus. I should have removed this one, but I would like you to understand the amount of non-human primates present in Fiocruz. In fact, they have specific areas to raise the animals, and obviously animals from the old world and the new world have their own peculiar characteristics in terms of being raised and handled so that they can be used, in fact, in many different researches.
Agora eu fiz uma coisa muito grave. Provavelmente, e não é que nós chegamos.
And here, the most important homage to the animals that are their attributions. All researchers must be remembered to that because of rhesus monkey and fascicularis. Probably you all followed the polio vaccination with the Sabin, which started in the 50s, with a vaccine that is attenuated against the 3 polioviruses. This number is being mentioned now by me, because some people contestate that, but I would say that from 1950 to now, about 1.5 million monkeys from the old world were used only for the polio vaccines. It's just for you to understand the reach of these animals in decreasing and curing diseases that in fact were a big problem to the human race. We have an obligation from the point of view of animal history, that is Rodrigo's obligation, and all of you working with animal research, to use all the forms and the ways to comply with animal ethics and welfare. So if you work with animal experiment, you need to know that the 3 R's are mandatory in the ethics policies to use any kind of animal, lab animal, since the small rodents of my friend Rodrigo and the big African primates. So we have important dating so that you replace and you reduce and you refine your experiments. All the experiments. Okay, now it's time for me to drink water.
Eu esqueci de tomar droga hoje. Isso é que dá ter plano de saúde.
With health insurance, we're immortal. That's important to clarify. Some surgeries, just 16, making us work and look alive. Continuing, there is no possibility that any of us be self-taught relating to experimentation with animals. All of the guides that are produced as norms should be carefully followed. So here we have the norms for the conduction of non-clinical or pre-clinical works, which are very up-to-date, and they encompass all the species that you could be working with right now. Something else that is essential inside our comprehension, especially economic comprehension, is for you to understand that any experiment that uses non-human primates, the involvement of a cost or price is based on whether the animals are SPF. So if we work with a rhesus SPF that is within these possibilities, you will spend about $8,000 or $9,000 for each rhesus. And that's a demand for the production of vaccines, drugs, neurobiological experimentation, you can only use animals that are SPF. And this cost of an SPF animal demands that the lab or the creation center be accredited. And this is another very serious issue. You need to have these credentials for the production and supply center. So with any kind of research that you conduct, for example, in Bio-Manguinhos, 20 rhesus monkeys are necessary. You would already start with a cost of $160,000 just to have these SPF animals. And that's a bit of the reason of why we have 58 protocols in 38 years of work. The lab was constructed in 1982, and once again it was expanded in 1998. Today the lab only has 420 square meters. We can maintain it in a much simpler way. It's a very big area, but the demand is that this vivarium of creation should have negative pressure, control for access, operational procedures, just like any small or big lab for experimentation would have. And this demands that you should spend your day, your whole life really, just to get a lab like this to be in the adequate conditions for working. This is a simple presentation of a lab, experimentation lab with primates. This involves specialized colleagues, the correct procedures, specific cages, monkeys that have already been screened for these diseases that you are presenting at the moment. For example, if it's Zika, they have to be serologically negative for Zika. Chikungunya, they need to be serologically negative for chikungunya. And you need to consider this for other pathogens. The lab consists, as I said, it has been here since 1982. We've been working for 35 years with many units. As I said, as Rodrigo said, production labs, development labs that make us have a great amount of activity. And this is the location by Avenida Brasil, surrounded by small units that always pollute our air. Here we have the INCQS, and this is our unit. Leaving from Avenida Brasil and entering, we have quick access here, then to LANIL, which is the neurovirulence lab. The studies we conduct involve the viruses. In the studies we can see if the viruses can create issues in the nervous system. These we need to identify if they are arboviral vaccines, yellow fever and dengue, mumps, rubella and measles, or other viruses that could have a neurological implication. So effectiveness studies and immunogenicity studies involving hepatitis A, C, dengue, Zika, parvovirus. So in our job we use primates. We conduct the intracerebral inoculation with a neurotropical virus or a virus that in these animals provokes a clinical condition that can be compatible with one that is presented in human beings. The main protocols, international protocols conducted, we started in 82 with Japan, polio, then Canada. We don't have to read all of this, you know what's going on here, but just so you can see that we have had an international presence based on our researchers who are no longer with us. Some are already retired, others have left the institution, but they gave us the opportunity during these years to participate and to obtain knowledge about a number of diseases, like, for example, working with the yellow fever vaccine. And this was a great start for all of these protocols. We also had Brazilian partners that participated actively with the neurovirulence lab. In 1985 we started, and then we never stopped. Oswaldo Cruz, UFI, bacteriology, and other agencies, they all worked with biomodels, non-human primates. So all of these protocols that you see here are actually conducted by a large group, including my friend Marcos Freire. Is he here today? No? So he asks me for the following: don't forget to mention that all the protocols that are conducted by you at LANEU should mandatorily be multicentric. There are many labs that are qualified to collaborate with LANEU for this protocol to move on. We are not totally independent to obtain things simply by conducting trials with animals and obtain results that are convincing. We need these collaborations with this big network of people. Some of you are here, and you need to know that you have to consider time, schedules, a lot of work. I know some of this work is very demanding. My colleagues are listening to me. We had about 600 days of work with no breaks for some of these trials. So within these 58 protocols, some experiments even lasted for 2 years. And I'd like to finish up with a little mural, people that many of you probably remember, and that were part of this trajectory. Of course, a tribute to the greatest experimental pathologist in history, Oswaldo Cruz. And some of the main actors in our story are portrayed here. Some of them, unfortunately, are not among us anymore. Thank you very much for listening, and I'll see you next time.
Muito obrigada, Dr. Renato.
Thank you very much, Dr. Renato, Rodrigo, for your words. Now we will move on to our pitching session. Let's take a small break for them to load their presentations. So we're going to take a small break so the speakers can organize everything here. So we're just going to take a small break before the next session, the pitching session. Advisor for our technological development, unfortunately, sorry, excuse me. Sorry, that's my mistake. He is in Europe. It's good for breaking the ice. His flight was canceled, so he will only be able to arrive after the event. He was supposed to arrive on Tuesday, and he had a number of issues with his flight, so he's in Europe. And he can't be here today. So we have Masarasawa, Vice Director of Technological Development. Also in the examining board, Eduardo Soares from Bio-Minas, CEO from Bio-Minas, also in our examining board, and João Miguel, who is an advisor for the Vice Presidency of Production and Supplies and Innovation of Fiocruz, representing Marco Krieger, who also could not be here. He is in Brasilia at a meeting, so I apologize for my mistakes here. And now we are going to start the first presentation by Ronaldo Sembio. You will have 5 minutes to make your presentations. So this is fast. You know the drill. So we also thought João Peroni from BNDES would be here in the examining board, but unfortunately due to his schedule he could not show up, and he will not be here tomorrow, so we will only have 3 people at our examining board, those who I introduced to you. Thank you for being present here, and we will start with Ronaldo's presentation, SemBio. Good morning, everyone. Good morning, everyone. My name is Ronaldo. Thank you for the opportunity of presenting SemBio. I'd like to start making these questions for when we work with immunobiologicals. Were they produced in the correct manner? Is the sequence of amino acids correct? Is the three-dimensional structure correct? Are they contaminated with other macromolecules? What is the degree of purity of this immunobiological? Are post-traditional modifications correct? Because usually these molecules are produced in mammals and need to be glycosylated and other modifications. The different lots, are they reproducible? Are the biosimilar identical to the original product? So to answer these questions, you need to go through analytical methods that are very specific and precise. In this case, I coordinate this, and this technique is the only one that can answer all of these questions. It is a mass spectrometry technique, and it can answer the questions in a specific way. So within mass spectrometry, we can analyze proteins, lipids, sugars, organic compounds, analytic method that is very powerful, precise, sensitive, and that can answer the questions that I made in the first slide. At SemBio, we have 4 mass spectrometry equipments that can answer these questions, and we can conduct a series of analyses for identification of molecular mass, fragmentation, imaging, analysis of lipids, and quantification. And in this equipment, we have AutoFlex, IOTrap, and other pieces of equipment. More recently, we acquired the mass spectrometrist called Solaris, which is cutting edge in Latin America. It is used normally for this kind of analysis, and this piece of equipment is available at SemBio for the users. It can conduct a variety of chemical analysis, especially fragmentations, and determine the exact mass with great accuracy. We also have the QTOF, which can analyze intact proteins and other elements. We also have equipments, MALDI-TOF that can be used, and the ion trap. All of these pieces of equipment are part of SemBio. The center is a facility that we are used to, just like those in the US. Users show the samples, they have the results, and pay for them. So this is done in a very professional way, and it is available for the scientific community. We have a partnership with Bio-Manguinhos, with Fiocruz, and a variety of researchers already use our facilities. They are our partners, and I want to show you that this exists. It is at UFRJ, it is available, and we are open to starting new partnerships and making this technology available, which will be extremely necessary, not only for advances in characterization of biopharmaceuticals that we will start producing here in Brazil. Thank you.
Falei quase 4. Não, é porque é muito claro. Aqui, ó. A televisão é muito clara, ele não aparece na TV. Aparece? Cadê? Não, só aqui. Aqui aparece, aqui esse também aparece aqui. Aqui não vai.
Agora tá indo mesmo.
Ela tá indo? Já que tá. Bom, vamos lá. Muito obrigado pela apresentação, Ronaldo.
Thank you, Ronaldo.
As perguntas vão ser ao final da sessão.
Will be in the end of the session. Everybody will present.
Obrigado pela participação.
Thank you for your participation. This is the certification. Anyone who presents here will have their certificate. Now, Juliane, come to your presentation.
Para a empresa Novas Ferramentas.
Gianni, for the company, do you have a company?
A UENF.
Okay, UENF.
Vai apresentar novas ferramentas
will present the novel immunochemical tools for differentiation of tuberculosis and non-tuberculosis microbacterial diagnosis.
Bom dia a todos.
Good morning, everybody. The title of my work is the development of immunochemical tools for the rapid diagnosis of non-tuberculosis microbacterial infections and tuberculosis, and we are working on the labs of UENF. Tuberculosis is one of the infectious diseases who kills the most in the world, especially in developed countries, and in parallel, mycobacteria is increasing worldwide, both in developed and underdeveloped countries. It's important to emphasize that mycobacteria from the AVO complex are more prevalent worldwide. It is important to have a differential diagnosis of these infections due to the clinical and radiological similarities, as the treatment for the infections is different. And currently the main diagnosis methods we have is bacilloscopy, X-ray, tuberculin, skin test, and they present the disadvantage of not being able to differentiate mycobacteria. We have the culture that is the gold standard, but it takes time for the diagnosis due to the slow growth of mycobacteria, and PCR, which is rapid, but we have sophisticated and high complexity equipment, which is quite expensive in low-income countries. As an alternative for the diagnosis, we have the immunochemical tests, which allows the identification of unique antigens or that are expressed differently from the bacteria. The antigen as identified as MTP is only secreted in mycobacteria, and it's used in the immunochromatography test for the detection of the antigen in liquid and solid cultures. The advantage is that it's a simple method with high sensitivity, but the disadvantage is that it identifies only the mycogenes of tuberculosis and not the other ones. So to identify new antigens, especially for the diagnosis of infections, our research groups developed and identified the antigen APA mode from Mycobacterium avium, and we produced monoclonal antibodies against this antigen. And we could check that the monoclonal antibodies produced only recognize Mycobacterium avium and not the Mycobacterium tuberculosis when compared to the polyclonal antibodies, which recognize both complexes. In 2011, we published a paper on this data, and this way we produced a kit, a sandwich ELISA kit, anti-APA, that is for the identification of APA antigens in feces of animals with paratuberculosis and in different cultures of MEAV, and we published the patent, and we recently published a paper on the use of this antigen as a biomarker, and we applied the sandwich ELISA for the identification of the APA protein in the supernatant of cultures, and we could observe that the kit only detects APA protein in the supernatant of the culture of the N avium complex for tuberculosis, and Mycobacterium avius and sus, and it doesn't identify APA for the Mycobacterium casaceae and Mycobacteria from the tuberculosis complex. So this ELISA identifies only APA in the M. avis complex. So the next step is to develop an immunochromatographic test to detect the antigen in culture as it was produced for MPT64. The production of a multiplex kit based on the detection of antibodies for anti-APA and MPT64 that we are producing this monoclonal antibody in the lab, and assess the use of this antigen in the diagnosis of mycobacteria infection ex pulmonary, as it was proven recently that MPT64 of Mycobacterium tuberculosis may be detected in these infections. And currently, we have a sandwich ELISA kit to detect, and we tested in different clinical isolated, and our interest is to look for companies to help us in the development of the kit. We have a patent currently that is filed, and this is my contact email. Thank you for the opportunity.
Tá direitinho. Faltou isso? Tá bom. Assim, Juliane, seu certificado. Obrigado pela apresentação e participação. Bom, oi? Foto? Pode ser. Cadê o... Tá. Bom, a próxima apresentação.
Next presentation is Thalita, from Thalita Souza.
Thalita Souza, da empresa aqui.
Do you have a company?
From
UNB.
Vai falar sobre fator de crescimento.
And she will talk about the growth factor.
Tudo certinho, apresentação. Sim, só laser point. Laser point é só para explicar. Eu não vou precisar ver, só direito. Agradeço a presença.
Thank you for your presence. Have a nice presentation.
Bom dia.
Well, good morning. My name is Thalita,
and
this project of production of PDGF from platelets using mammalian cells is a collaboration with the Nucleus of Cellular and Molecular Therapy of USP, the Medicine School of USP. I work with them in the lab and development of some work in this project at the University of Brasilia. The first point to be addressed here is to know what is PDGF and what would be the main actions of this glycoprotein in some systems. First, PDGF is part of a cascade of blood coagulation and cicatrization, so it may be used basically for repair processes in the tissue and regeneration. Within this area, it can be part of regeneration of tissue or periodontal regeneration, cell differentiation, ocular neovasculation, coming from the principle that it is a chemiostatic substance, mutagenic and angiogenic, and it also has this angiogenic action. And what does diabetes has to do with the PDGF? Diabetes mellitus is a metabolic syndrome characterized by many diseases associated, and one of them is the peripheral neuropathy, affecting 50% of patients with diabetes. It's important to mention that 90% of the Brazilian population has diabetes, both type 1 or 2, and we believe that about 20 million people will be affected by diabetes in 2045. Currently, we have 851 million patients in the world with diabetes.
Com relação ao governo
regarding
federal,
the federal government,
foi-se gasto 24 bilhões
they
de
spent
dólares
$25
no
billion
tratamento
in the treatment of diabetes.
Bom, para se desenvolver ou para
develop or to study better
a produção desse fator
the
de
production of this growth factor, we created this approach and we used human kidney cells with antivirus carrying the construction of PLPDGF. And you have to remember that these cells are adherent cells, so we first had the process of building our strain and then establishing the strains in adherent bottles, then followed by the increase of scale for systems adhered to microcarriers and the characterization of our glycoprotein by SDS. It was followed by Western blotting, and then the quantification with the ELISA method with a commercial kit.
Depois, seguido, então, de alguns ensaios
followed by some in vitro and in vivo assays that I'll show later. So this is our first construction, the first strain we created. Then we followed with a cell culture in bottle and followed by spinners and wave. And you have to remember that currently we produce cultures in 1 liter of culture medium. Then we went through the purification stage. By affinity, we used Actor Purifier, and here we have the SDS page proving the presence of our protein and also Western blotting. And also here we have the biological activity of our extract proving by timidina tritiated assays the presence of this activity. Some preliminary in vivo assays evaluating the cicatrization of wounds with PDGF, and these are the main goals, the main steps from now on. Thank you.
Obrigado pela participação. Próximo candidato é Patrícia.
Next candidate is Patricia. Patricia Prank.
Qual a empresa?
What is your company?
Da Federal do Rio Grande do Sul.
Okay, from the Federal University of Rio Grande do Sul.
Eu vou pegar minutinho e meio do Ronaldo, que ele não
I'll take 1.5 minutes from Ronaldo. Anyway, good morning, everybody.
Opa, não começou ainda. Quero me adiantar. Ok. Bom dia, meu nome é Patrícia Prank, eu sou professora de hematologia da Faculdade de Farmácia da URGS. Estou aqui para apresentar um projeto que, na realidade, eu chamei aqui como subprojeto, o Recupera-Med, mas ele já faz parte de um projeto que eu chamo de Projeto Andar, que nós já trabalhamos no laboratório há cerca de 10 anos nesse projeto. Eu fiz doutorado nos Estados Unidos há cerca de 15 anos eu trabalho com células mais recentemente, há cerca de 10 anos, nós introduzimos a nanotecnologia e começamos a estudar mais a lesão de medula espinhal, que é
the lesions of the spinal cord with this project on there, and then we had the sub-project that is Recupera-Med. We also have the Institute of Research in Stem Cells, and our initial focus was to use stem cells for the hacromedular cells. We have great access to the website and patients, and we have lots of access every month. And in the Institute we have Drauzio Varela, Luiz Fernando Veríssimo, Cristina Ranzolini,
Caroline Falcão, many researchers and scientists and business people supporting the project. And I would like to remember that Dr. Maiana Zatz from USC were one of the actors, along with me, to approve the law for the stem cell research in Brazil. We have this law, it was approved in 2005 to use embryonic stem cells. And why am I saying embryonic? Because I would like to have embryonic stem cells to help patients. But then we started dealing with pharmacy and medication, and to my surprise, although we have more than 10 papers publishing showing stem cells as a good use and the best approach for medullary lesion, we had some other medications that was much better, and it was a big advantage because it's much easier to use a medication than a stem cell. And this is what I would like to show. Nanotechnology was introduced as the nano delivery because for 10 years we've worked on that in the lab. Needless to mention that nanotechnology over the years is increasing a lot in terms of publication and prices and amounts that the formatting agencies both in Brazil and abroad are dealing with that.
The spinal cord injuries, they confine people, a lot of times young people, they are confined to a wheelchair. In the world, we have around 2.5 million people affected by this, about 130,000 new cases per year. In Brazil, this is very alarming, 40 new cases per year per million of inhabitants, 6,000 to 8,000 new cases in Brazil of people who are confined to a wheelchair. And we know there is no cure for this. There are few medications available, few treatments to improve their quality of life. They say that any improvements for them help them, even just controlling their sphincter, anything like that would be great for them. Some results with mice have already showed improvements, which would be about equivalent to this. The patient can control their sphincter when before they were not able to. So we're trying to get these improvements, get these resources to accelerate these results. Laísi, this is Laísi, I met her. She supports our work because I'm starting a partnership with Miami University. She was treated there. She's a triathlete. She started this treatment with stem cells at the University of Miami a few years ago. So I can't reveal the name of the drugs, but the good news here is that these are drugs that are already used today. We are just presenting a new pharmaceutical formula. And what do these drugs show? What I'm going to show here in this graph is that 0 means no movement in the mice, and 21 means the maximum amount for normal movement. We always work with this control system, so if the mouse suffered this injury or partial injury, one that has spinal cord injury, and our treatment, there's always a spontaneous improvement in all of the mice. For example, this group of mice went from 11 to 16, so it's not perfect, but it's a significant improvement. In the second treatment with other kinds of drugs, this was a lot more visible. We worked with the brute form of the drug. Now we can see we're showing its efficiency. We're working with derivatives, purified forms, and we have partnerships with a lot of countries around the world. Miami University will be our partner. If we think about clinical trials, maybe we can do them here in Brazil, but there they have everything ready to conduct these kinds of studies. So the objectives are to continue studying, developing medication inside the hospitals, to do this business to business in the pharmaceutical industry. If anyone in the industry wants to support us, that's fine, and to improve the results. So maybe not obtain a complete cure, but improve their quality of life. That would be great. So to do this, we are doing molecular modeling. Oh, I don't have any more time. And the advantages of our products, sorry, I didn't have time to finish, but thank you. This is our team. And now, Ana Carolina from the CELP software from Unicamp to present the CELP software.
Os índices do meio, que você lá do meio vai travar a apresentação. Então, voltar, só se por acaso passar mais ou menos esse aqui, se precisar voltar, você volta.
Tá bom, então aqui.
Não aperta meio.
Só aqui, né? Tá.
Não
tá indo, né?
Primeiramente, bom dia a todos.
Good morning to everyone. My name is Ana Carolina. I am a Unicamp student and I'm here to present the SELP software counter of erythrocytes, leukocytes, and platelets. So around the world we have around 7 billion inhabitants. 50% of this population lives in poverty and 25.6% live in extreme poverty. So we are speaking about people who live with less than $1 per day. So let me ask you, if these people don't have money to eat, how can they take care of their health? On the other side, we have a different outlook. People who live in countries that provide some kind of health, total or partial, for the population of the country. Here in Brazil, we have the unified health system. 70% of the population depends on the unified health system in Brazil. When we speak about health, it is important to highlight the medical diagnosis. 70% of medical decisions are taken based on lab exams. The hemogram is the most demanded one. Currently, the hemogram is made with 3 methodologies, the manual method and the automatic method. The manual method has a low cost, low speed, and low accuracy. Automatic method has high cost, high speed, and high accuracy. So you must be asking yourselves, how high is this cost? Before I started this project a while ago, I called a lot of representatives of hematological equipment, and I saw that this price is about 25,000 to 90,000 reals. So why can't we create a third methodology with low cost, high speed, and high accuracy? And why can't we do all of this through a simple computer? The answer is yes, we can do this through the SELP software. When it is concluded, it will be a computer program based on digital image processing techniques, where its innovation is counting simultaneously the 3 kinds of blood cells in the pathological and non-pathological state in the Neubauer chamber. And here I have some images of how this algorithm works. Our potential market in Brazil, we have about 200 million inhabitants, where 70% of this population totally depends on the unified health system, the SUS. So it's an essential system. Any methodology that can reduce costs is always very welcome. In the context of private hospitals, they are 1/3 of hospitals in this country, so we can use this methodology as a method to confirm this. And since we are speaking of reducing the cost of methodology, automatically the price of my service will be lower. It's more accessible to less favored populations, indigenous populations. And in this idea of reducing the cost of the methodology, we are indirectly influencing entrepreneurship, especially young people who are finishing college now, want to be entrepreneurs, but they think it's expensive to pay for equipment. That's over 25,000 reals. Our business plan would be to start sales by e-commerce. We are thinking of about 5,000 reals, which is a lot cheaper than conventional equipment. And in this first year, we would like to reach 3% of the market. This is my team. I have a biomedicine degree. I'm doing a graduate program in electrical engineering, and these are the people from my team. They are all masters or PhD and they are professors at state universities. Our objectives for 2019 are to develop the algorithms that will count these cells in pathologic and non-pathologic states, test them in terms of accuracy, specificity, and sensitivity, and in different kinds of computers. Our objective here at the Innovation Hub is to look for intellectual and financial partnerships that can help us to validate this methodology when it's finished, and that can help us to put this product in the market, because that is how we can get our objective of helping people with low income to have high quality health. Thank you so much for your attention.
Só pegar seu certificado. Passa o certificado. Obrigado pela presença.
So now that the presentations have finished, we are opening this up to questions. If anyone here has questions or comments, feel free to make them. Any questions, comments, issues? First of all, of course, the examination board is the examining board is the priority. Any questions? Any considerations? Well, congratulations to everyone. First, you need a lot of courage to do these pitches. You're exposing yourself. You need to know what you want, what your objectives are. So congratulations to all of you who were willing to do this. Thank you for your time. It's so important to be here and look at new ideas, because I think our objective, we all have the same objective. We want to work for the population's health. First of all, I'm not going to make comments on the presentations. I don't know if this is the moment, but I'm going to make a very specific question to Patricia. These drugs that you cannot reveal, but that they are already in the market, are they synthetic or biological? What are their bases? Well, they are drugs that we use in other diseases. The innovation is using them for spinal cord injury and the new formulation we are creating. And will it be necessary to conduct clinical trials for this new therapeutic use? Yes, I think so, because it's different information, but it's already very well accepted for a disease, and I think the clinical trial will not be difficult. Andrea, are we going to have a moment for comments later? Okay. So generally speaking, the presentations were good. I thought in most presentations there was a lack of a business plan, a structured business plan was missing. Maybe that's just my perception because it's my area. I look at the business part of things, but I thought that was lacking. But in general terms, I was able to understand the proposals, the objectives, and what you're looking for here. And this allowed us to be able to analyze the viability, how this will be developed, if it's worth investing, and if it's possible to work with a partnership to be able to launch these projects. So congratulations. I hope everything works out tomorrow as well. Well, first of all, let me stand up. I want to thank everyone who made a presentation. For those of you who know me a bit, I make my life doing this. I listen to people's pitches. So I'm kind of trained to understand and listen and see what you want to say, what you should say, and what you shouldn't say in a pitching session. In general terms, I thought there was a lack of a business model. I think you should think of a business model. And there are many ways to do this. Some people have been through our program, so they know how to do this, right, Ana Paula? You know about this very well. Something that's very important and that most projects presented forgot about it, let's say, and it's essential for us, in the examining board and for potential investors who are here in our audience is the team. Most of you did not speak of your team, and the team is essential here. You need to speak about who you're working with, who's part of your team. So if any of you will present tomorrow, please add this section about your team. And review the market issue. Who is your market? Who are you selling this to? You use too many technical slides, so you should reduce the number of technical slides. Just use the direct information, the basic concept, make it fast and focus on the market, how you're selling this, how you expect to sell this. And you might not even have the correct model, but it's good to at least have a model to present, and then maybe somebody will review this and give you some suggestions about what can be done to improve the project. But in general, these are my comments, but the projects are great. I enjoyed it very much. Well, in name of the Vice Directory of Technological Development, I'd like to thank everyone's participation, the speakers as well, and I missed this. I thought you were lacking the same issues that João and Eduardo mentioned. We know how it's difficult to work on development here in Brazil. It's a constant struggle. So I'd really like to congratulate all of you for this initiative, and we are here. We will be here tomorrow as well. So any other observations, comments? Thank you for your presence. We'll see you tomorrow for the next pitching session. Thank you for your presence.
Obrigado.